Okay, so we start. If we were to have science in newspapers, which we have sometimes, not very often, but in a newspaper like the Times, we could have found a few years ago a title saying, Rimonabant, new anti-obesity miracle. Have anyone heard about Rimonabant? Yes? Okay, what is it? So you know about it, but you don't know what is it? Rocío?
It's a drug.
It's a drug? Okay, and what's the purpose or the effect?
I don't know exactly, probably makes you don't absorb the lipids, probably.
That one is Orlistat. Okay, that was a safe option for all of you, just to say it helps against obesity, which is written there. So yes, it helps us. Yes, Ian?
It blocks the receptors of cannabinoids.
Oh,
Cannabinoids.
a good student there. Yes. So we'll see. So I have this first slide here because Rimonabant is the main culprit of researchers studying the relation of endocannabinoid system, inflammation, and adipose tissue. Rimonabant opened the door to this kind of studies. So a bit of chronology. 1994, it was synthesized, okay? It's a chemically synthesized molecule. It's not an endogenous molecule or a natural molecule. 2004, it was started to know scientifically. What is the meaning of this? We scientists could find articles about Rimonabant and obesity in PubMed from 2004. And in 2006, it was started to be sold in the European market, okay, for the treatment of obesity. A very important thing here, or an anecdote, which is quite funny, is that Rimonabant was studied as a drug to stop smoking, okay? And they start the clinical studies to check if it was good to stop smoking, and they realized that the patients, the volunteers, were reducing their weights. So at some point they decided to go more for the obesity market, and they sold it as an anti-obesity drug. But unfortunately, in 2008, 2009, was withdrawn from the European market. What is the meaning of this? It means that we can't buy Rimonabant right now, okay? Because it increased the suicidal intentions of the people, okay? And as you may know, obesity people tend to have depression, tend to have close to these sensations, to these feelings, so obviously it's something not very good. But to show you how this story begins, till 2003 we had 70,000 articles about obesity, about 7,000 about cannabinoids, and only 23 about cannabinoids and obesity. I'm not talking about inflammation or adipose tissue, just cannabinoids and obesity, only 23. From 2004 to the mid of 2008, there were 323 articles about that, okay? So scientists realized about the possibilities of the connection of the cannabinoid system with obesity. Okay, so now an easy question. What's on the background? We. Okay, something more formal. Or marijuana, but too formal, cannabis sativa. Okay, so cannabinoids, endocannabinoids, are cannabis sativa links to the receptors of cannabinoid system. Okay. So the endogenous molecules are anandamide and 2-arachidonoid glycerol. Both of them join to the two main cannabinoid receptors, CB1 and CB2. And I have to come here. And they are related to arachidonic acid. So as you can see here, this is the molecule of arachidonic acid, and this is anandamide and arachidonoid glycerol. So they are practically the same, as you can see. This one is a chemically synthesized cannabinoid ligand. Okay, so you can see that it is quite different from these other two. The expression, okay, where do we have receptors of this endocannabinoid system? We have them in practically every tissue of our body. Mainly in the brain. That's why there was this problem with rimonabant, because it acts centrally and it acts peripherally, but mainly centrally. And we cannot control properly the effects of the central receptor. And then stomach, and then adipose tissue. Okay? The third tissue with the highest amount of CB1 receptor was the adipose tissue. Okay, action. Stimulate appetite, relaxation, sedation, protection against acute and inflammatory damage. So it's there already, inflammatory damage. Help to forget aversive memories. Okay, this is more to the social part of the marijuana. So mainly for us, control of energy intake. Endocannabinoids lie within the area of what we call orexigenic factors. What is an orexigenic factor? Yes? Yes. Appetite mainly. So yes, so anorexigenics are the ones that reduce our appetite, and orexigenic are the ones that stimulate our appetite. What receptors do we know about endocannabinoid system? CB1 and CB2. And also we can talk about two other putative receptors, which are TRPV1 and GPR55. Why do we call them putative receptors, and why do we consider them as cannabinoid receptors? Okay? How is your knowledge of pharmacology? Not very good? Okay. All right, so in the bilayer of the external membrane of the cell, we have the receptors, okay? And these receptors act like a keyhole and a key, so the molecule has to link or join perfectly to the receptor, and then it will produce an effect, okay? So what we have seen is that anandamide and 2-arachidonoid glycerol, which are endogenous ligands of CB1 and CB2, also join TRPV1 and GPR55 and produce more or less similar effects. So that's why we have considered them as other cannabinoid receptors. Okay? Is it clear? Yes? So very briefly, CB1, strongly expressed in the brain, okay, mainly a central receptor. It stimulates appetite. CB2, on the contrary, is more a peripheral receptor. And it's expressed in immune cells, okay, so relation with inflammation. And it has an immunomodulatory role. TRPV1 is expressed in several tissues, and it has a crucial role in thermal hyperalgesia. So someone said over there, control of temperature. So it's here. And it's also involved in satiety and gastrointestinal motility and in adipogenesis. Okay, production of adipocytes. And finally, GPR55 is also expressed in several tissues. Recently, it has been shown mainly to be expressed in the brain, and it can be used for the treatment of inflammation and pain. But this is probably the newest receptor, so it is still under a lot of study. And it has been called in some articles as CB3, okay, the third cannabinoid receptor. Although apparently recently as well, it has been discovered one in the hippocampus, which is probably going to be called CB3. So maybe this one CB4, or we'll see. So functions of the endocannabinoid system. We have mentioned a few, but this system is very important in our homeostasis. So it controls energy balance, it controls stress recovery, it controls food intake, metabolic homeostasis, vascular responses, endocrine functions, neuroprotection, and nociception. You know what is nociception? Pain, okay, the feeling of pain, and immune modulation. If you don't understand anything or you want to ask anything, you can do it at any point of the talk, okay? You don't need to wait to the end, because usually you forget or you just get sight by the end of it. So if you have something to ask, do it when you want. Okay. So back again to rimonabant, just to explain a little bit why it's important, this molecule, to us. It's a CB1 antagonist, okay? Rimonabant is the specific name, and Acomplia is one of the commercial names, although, as I said, it's not commercial anymore. So what is happening? There is an excess food intake, there is a situation of obesity, and that is going to overactivate the endocannabinoid system, right? So that will stimulate appetite, as we have seen. But we don't want that. So CB1 is blocked by rimonabant, okay? So we stop that area of the endocannabinoid system, and we produce a decrease in food intake in the brain, a decrease in food intake at the gastrointestinal tract level, and at the adipose level, decreased waist circumference, decreased insulin resistance, decreased triglycerides, increased HDL cholesterol, increased glucose tolerance, and increased adiponectin, which is an adipokine related with anti-inflammatory effects. Okay? So I'm going to show now some statements from different articles to show the relation of the endocannabinoid system with inflammation and obesity. First of all, we have seen that one already. The endocannabinoid system is overactive in obesity. Okay? The activation of TRPV1 prevents adipogenesis, production of adipocytes, and obesity in 3T3L1 pre-adipocytes. Sang et al. From 2007. I have there the author because I personally wouldn't say obesity on a cell line. Okay? We can talk about obesity on a human, and sometimes, although it's probably not the best word, in animals, but not in cells. They don't get obese. Okay? Endocannabinoids and synthetic cannabinoid receptor agonists are immunosuppressive and anti-inflammatory. They have been related with colonic inflammation, well, with several diseases related with inflammation. 3 of the receptors that I have mentioned previously are highly expressed in immune cells. As you can see there, in monocytes and leukocytes, TRPV1 and GPR55 are highly expressed, and we will see later about CB2 as well. And finally, and very important, adipose tissue is infiltrated with macrophages in obesity, and activated macrophages synthesize endocannabinoids. Okay? Any doubts so far? Are you already sleeping, or did you get more or less? Okay. So in most of the talks of the course so far, you have seen a wide introduction about the theme. I'm going to finish more or less right now my introduction, and then I will start properly with what it was the research project in which I was involved, trying to get more answers to this relation. Okay? So first, something you have seen already, obesity. How do we go from that muscle man to the obese one with an impaired energy balance, in which we have at some point mild and chronic inflammation. So we want to understand a bit of how that inflammation is connected with obesity and with the endocannabinoid system. And also we want to know what is the relation of the endocannabinoids with pre-adipocytes, adipocytes, and macrophages, okay, which are the 3 main components of what we call the adipose organ. So the hypothesis is that the endocannabinoids have an anti-inflammatory action in white adipose tissue, modulating in particular the production of inflammation-related adipokines, and thereby protecting vascular function and insulin sensitivity when adiposity is excessive. Okay? Urchi? Everything, yeah? Right, so we conducted these experiments in cells. Sembio cells are human adipocytes, primary culture of human adipocytes, okay, obtained from volunteers. And SEBS cells are human adipocytes as well, but they are obtained from a disease, a human disease that produces obesity very early in childhood, okay? So it is not exact, it is a good way to check ideas about obesity, but it's probably not the closest model to most of the population. Okay, so Sembio cells is properly a primary culture. But we use both, but just to try not to confuse you too much, I will show mainly only the Sembio cells results and just a couple of the SDBS results. Okay, so there you can see a standard PCR gel in which I have a squared, red squared, sample number 5, which is monocytes. Okay, showing that CB2, TRPV1 and GPR55 are highly expressed in monocytes, in immune cells. And you can see other cells there. This is the protein expression of CB1, which surprisingly it's quite high as pre-adipocytes, and as we differentiate them, the protein is missed, and then as they are mature adipocytes, it's back again. Okay? And this is very important of why I'm only showing the Sembio cells results. The difference of expression of the cannabinoid CB1 receptor in Sembio appears much earlier. It is much highly expressed than in SEBS cells. So that makes it very difficult to get results and to understand them. All right? To try to solve that problem with SEBS, we made electroporation. Okay? Anyone have heard about electroporation? Anyone have heard about gene transfection? Yes? Okay, so Oxford Brookes, what is gene transfection? Anyone? No one? Okay, so if Gabi, you want to say something? No? Okay, so if we have a gene which is very interesting for us, and either we want to block the expression of that gene to see very well or to check very well what is the effect, okay, we can knock down through gene transfection that gene, or we can overexpress that gene to get an overreaction, right? So as the gene of CB1 was little expressed, low expression in these SGBS cells, we were thinking of increasing with electroporation. Adipocytes are a cell very difficult to transfect because of the lipid content. So we have to use strange ways. So I'm going to explain very quickly what is electroporation. Electroporation means we give an electric charge, a high electric charge to the cell, which in many cases will kill the cell. Okay? But what it does is, because it's quite hard for the cell, it opens pores in the membrane, allowing DNA or RNA to get into the cell, and then as it gets to the nuclei, it will express the gene that we want, okay? Or cancel the expression of the gene that we want, right? Everyone understood? Yep. So it is very difficult to transfect adipocytes. So as you can see there, we transfect the adipocytes and we obtain very little. The green areas mean we have been successful. So as you can see, most of the photograph is in black, so we haven't been very successful. So we tried something else. We tried to transfect as pre-adipocytes and then differentiate them. As pre-adipocytes, they are much more stronger, so we got a very good transfection as pre-adipocytes, but as they differentiate, that expression is lost. And finally, we got another idea, which was transfect them in day 2 when they are already with the differentiation cocktail. Okay, this is something many of you may not know. Adipocytes, well, the cells that we work as adipocytes are previously pre-adipocytes, and we have to give them some substances in order to differentiate them to adipocytes, okay? And that takes around 7 days. So on day 2, we transfect them. We got very successful. And as they mature, it was quite successful. But finally, we decided not to go for that way, and we took the same cells as we saw the expression was quite high in them. It was a safer approach. So as you can see there, the expression of the gene, it changes as they are starting to differentiate. So as pre-adipocytes, practically no expression, and then almost 600-fold expression, and decreases, and then increases a little bit as they are mature. This is from another group. CB2 is also mainly expressed in the differentiation period, and TRPV1 the same. Okay, so now to something that more or less half of the audience already have heard me talk about it, hypoxia. So obviously, as I was in Liverpool, we checked about hypoxia. And CB1 showed a decrease with hypoxia, and TRPV1 showed a decrease as well with hypoxia. For the people that have not heard me talking about hypoxia, I'm going to explain briefly. So hypoxia occurs in ischemic injury, wound healing, and solid tumors leading to extensive metabolic changes. Right? So you can see the arterial blood in normal conditions, 104. General tissue oxygenation, 40-50. Okay, so that's the value of the general tissue oxygenation, 40-50. There you can see some pathological situations in the spleen, thymus, retina, brain, tumors. And what about adipose tissue? In 2007, Ye et al. And Brahimi and Poesegu showed that in lean people, the white fat had 48 millimeters of mercury of oxygen, okay? But in obese, we got down to 15, right? So almost a third of the quantity we had as lean persons. Okay, so a big change in the quantity of oxygen. Another thing scientifics saw was that blood flow to white adipose tissue is not increased in obese subject. In contrast to lean subjects, blood flow to white adipose tissue does not increase postprandially after lunch in the obese. And also large adipocytes are further from the vasculature than the normal diffusion distance for oxygen, which is 100 micrometers, okay? And the large adipocytes are 150 micrometers of size. So obesity do not increase the blood flow. Okay, as you can see here, these are the adipocytes, these are the blood vessels, and this would be where we have this oxygen going to the cells. And as we increase our adipose tissue, we increase the number of adipocytes and we increase the size of these adipocytes. So as you can see, there is the blood flow, it's kept the same, but as they grew bigger, it is less quantity, and the oxygen have more difficulty to get to the cell. Okay, so other experiments we did. We are talking about receptors, okay? So let's check about different agonists and antagonists of these receptors. What is happening, okay? So we have explained briefly here about the receptors, about pharmacology of receptors. So who knows what is an agonist and what is an antagonist? Someone? Maybe someone working in pharmacology? Urchi? No? No one knows, or no one wants to answer?
An antagonist doesn't consider even or the connection or whatever you want. And an agonist, it
Okay. Yeah, more or less it would be that. An agonist linked to the receptor, and produce the effect expected from that system. Okay? An antagonist, usually it blocks the receptors, so it doesn't allow an agonist to link and produce the effect. Okay? And now increasing the difficulty, have you ever heard about inverse agonist? Okay, so in a very simple way, it would link the receptor and it would produce a different effect from what is expected. Well, not different, an opposite effect from what is expected from an agonist. Okay? So we tried all these molecules. We wanted to cover agonist, antagonist, of CB1 and CB2 receptors. Okay, and also the two endogenous ligands, and high dose and low dose. We did all this. We measure all those genes with all those molecules, but as you can see, most of them are showing no effect. So we focus on 3, but I'm going to show you just the last one, GTE 907, which we published as upregulation of the expression of inflammatory and angiogenic markers in human adipocytes by a synthetic cannabinoid. So you can see there is expression of CB1, TRPV1 in these cells, but we couldn't find of CB2. So we only measured CB1 and TRPV1. Okay, and you can see there is an upregulation of these genes. We measured the genes that you have seen in the table earlier. We measured some inflammatory related genes and some angiogenic related genes. Okay? So who can tell me out of those 6 which are inflammatory and which are angiogenic? Okay, Sonia? Okay, I'm going to save you the hassle. Till here, they are inflammatory. Okay, the first 4 are inflammatory related agents, genes, IL-6, interleukin 6, MCP1, monocyte chemoattractant protein 1, TNF-alpha tumor necrosis factor alpha, IL-1 beta, interleukin 1 beta, and the other 2 are related with angiogenesis, okay? The formation or production of blood vessels. Vascular endothelial growth factor, VEGF, and angiopoietin like 4 protein ANGPTL4. Okay, so you can see the effects are quite pronounced, are very important, mainly in the inflammatory ones. IL-6, it gets to 50 and almost 60-fold change. We checked when it was the highest effect. We saw that it was at 20 hours instead of 4 hours, so it's more like a prolonged effect more than an acute effect. Well, you can see with the genes, it shows a similar picture than previously. We saw that the expression of interleukin 6, the production of interleukin 6 protein was increased as well. Okay, and then we wanted to check if these effects were going through the CB1 and TRPV1 receptor, or they were going through another way. So AM251 is an antagonist of CB1. Okay, so you can see there is a decrease in the production of CB1 and there is a significant decrease in TRPV1. And the effects that we were seeing before were completely reversed. Okay? So it seems that it was working through CB1 receptor. Okay? AM630 is an antagonist of CB2. GT907 is an inverse agonist of CB2. Okay? So we should get similar results. And as you can see there, we got additive effects. Okay? We increased even more CB1 expression and TRPV1. And the same with the genes. Although only 2 were significant, but there was an additive effect as well. How about pre-adipocytes? The effect was the contrary. Okay? CB1 and TRPV1 were least expressed. And the same with the genes. Okay, so we saw there was a difference between adipocytes and pre-adipocytes, and we decided to check for the crosstalk between the 3 types of cells that I was talking earlier: macrophages, pre-adipocytes, and adipocytes. And we checked for all the possibilities: macrophage with pre-adipocyte, macrophage with adipocyte, pre-adipocytes with macrophages, pre-adipocytes with adipocytes, well, all the combinations, and only 2 of them showed significant differences. Pre-adipocytes with macrophages up 16-fold, okay, the CB1 expression, and adipocytes with macrophages down 5-fold. Okay? How we did this crosstalk? Some of you have been in a seminar I have given about these co-cultures, but most of you probably don't know about co-culture. Do you know something about co-cultures? No? Okay, so there are several options. We can have the cells, 2 types of cells within the same plate, okay, which is where we grow the cells, but that's quite difficult, okay, and many times it doesn't work. So one of the easiest options is used activated or conditioned media. So how did we do that? Okay, I'm pretty sure right now you still don't understand anything, so I'm going to explain. We have 2 plates, okay, with 2 types of cells. Let's say pre-adipocytes and adipocytes, okay? We grow them. So we get some factors in the medium because they are pre-adipocytes or because they are adipocytes, okay? Everything agrees with that? Yes? And we decide one is going to be the main type of cell. Okay, so this time we decide pre-adipocytes are the main one. So we take with this plate, okay, we change the media, and we put 850 microliters of the pre-adipocyte media, and we take 150 microliters of the adipocyte media. Okay? So in theory, the proteins and the adipokines that the adipocytes have been producing, we are going to put some of them here, and we will see somehow how it would be to have together pre-adipocytes and adipocytes. Okay? So that's what we did here. Okay? Control, it's the main type of cell, just alone, without anything. And conditioned media is media from the other type of cell, but without cells being grown there, okay? Because pre-adipocytes media has some factors in it already, and adipocytes media has some other factors. So that may also produce a change. And we just want to see the change of the proteins that are being produced by the adipocytes. Okay? So CB1 and TRPV1 were upregulated with the macrophage media, okay, in pre-adipocytes. And as you can see here, this macrophage media produce a huge inflammatory situation in these pre-adipocytes. Okay. 100-fold change, more than 110. Interleukin 1 beta, almost 1,300-fold change. Okay, so huge changes. What about adipocytes? In adipocytes we saw the opposite, a decrease in CB1 and an increase in TRPV1. Okay? But again an inflammatory situation. So in this case we didn't know if it was only going through TRPV1 or maybe other receptor. Inflammatory related agents. Okay, well, you know that the bad things about obesity are the inflammation that comes with it. That is what it produces: insulin resistance, atherosclerosis, and osteoarthritis and other diseases. So we checked for TNF-alpha with GTE, the inverse agonist we were using before, and we saw again an increase in CB1 and TRPV1 and an increase in the inflammatory and the angiogenic markers. Finally, arachidonic acid. Okay, we have shown at the beginning that it's very important, arachidonic acid, because it's the precursor of the endogenous ligands of the anandamide and arachidonic glycerol, which are over here and over here. Okay, so we decided to check for fatty acids, what it was happening with fatty acids and mainly arachidonic acid. So we checked for arachidonic acid, eicosapentaenoic acid, and palmitic acid. And you can see that CB1 and TRPV1 were increased, were upregulated with arachidonic acid, which makes sense because it's the precursor of the endogenous ligands. We also obtained an upregulation in the inflammatory and the angiogenic markers. And summarizing the results, I have selected only the most relevant results of this research project. So the conclusions would be about JT907, a cannabinoid CB2 receptor inverse agonist, upregulates the expression of inflammatory and angiogenic markers in human adipocytes. Macrophage condition medium upregulates CB1 and TRPV1, and macrophage condition medium upregulates in adipocytes only CB1, and TNF-alpha and arachidonic acid treatments upregulate the expression of both of CB1 and TRPV1. So to finish, a little about pharmaceutical industry. There are 4 figures there. Can anyone tell me about them? What is 12 years? 12 years is the average time to get from synthesis of a molecule to getting that molecule into the market. Okay? The next one, 1,000 million euros. Okay, so if we get a molecule from the very beginning to the end, okay, to commercialization, that uses or spends 1,000 million euros. Do you want me to write the zeros here? Or 3? 10,000? Could be both of the options, people, experiments, but not other possibility. In order to get one molecule in the market, we try 10,000 molecules. Okay? And the last one, 3 out of 10.
Only 3 drugs out of 10
Are what, sorry?
effective, they work.
Not exactly. No.
I think so 10 years is a patent for the drug, and only company has only 3 years to make money.
It has to do with money, but it's not that one. At the end of the room, people is very quiet. How about medicine students? Master students on that side, you are very quiet, apart from Suyen. Three of, yeah, more or less. So, of 10 molecules that are commercialized, that are in the market, only 3 of them produce enough money to cover the cost that we have produced.
We don't believe.
Well,
The richest company in the world are pharmaceuticals. So, you know what?
well, yes. I'm not going to say anything because obviously I got that from. Yeah, so could be biased, but that which is true in that sense is that the time of the patents are being reduced. So many pharmaceutical companies are reducing dramatically the investigation, innovation, development, and research in new drugs. So the connection with global health and individualized nutrition has to do with the figures we have just seen. Pharmacotherapy in obesity is very complicated. Rocío was mentioning Orlistat a few minutes ago. Right now in the market, we can only talk properly about one drug to treat obesity, which is Orlistat. Like 4 years ago, there were 3, sibutramine, rimonabant, and Orlistat. And sibutramine and rimonabant, both of them were withdrawn because of the side effects. What is the problem? The problem is that, as we have seen in many talks already, obesity is multifactorial. So with drugs, it's very difficult to find the right place, the right gene where we want to target our molecule. So it's very complicated to avoid side effects, important side effects. That's why individualized nutrition or other therapies could be very important, and maybe even individualized pharmacotherapy. So I would like to finish thanking Obesity Biology Research Unit in the University of Liverpool, where I did practically all this work, which was supervised by Dr. Chen Bin and Professor Paul Traherne, and was funded by the BBSRC, Biotechnology and Biological Sciences Research Council. I would also like to thank our sponsors, the University of the Basque Country, the IUNS, the International Nutrition Society, the University of Navarra, and the Cátedra Tomás Pascual, which has provided a good economic support for this course. And finally, but not least important, I would like to thank you all for your attention. Any questions?